DNA is divided into genes. We have about 20 to 25,000 genes and about 2 meters of DNA in each cell. DNA is made up of nucleotides. One nucleotide consists of a sugar, a phosphate, and a nitrogen base. The nitrogen bases are called adenine, thymine, guanine, and cytosine, referred to as A, T, G, and C. The genes in our DNA are the basis of life because they are the recipes for building the proteins our bodies needs to survive.
Transcription is the process in which the DNA sequence is copied into messenger RNA so that it can be used in translation and then eventually make protein. It starts in a phase called initiation, when the enzyme RNA polymerase binds to the promoter region in the DNA. This binding causes the two strands of DNA to unwind and eventually get pulled apart. The second phase is called elongation, and it starts with one strand of the pulled apart DNA being used as a template. The messenger RNA is created as a complementary copy of this DNA strand, which means that the copy consists of the opposite nitrogen bases of the template. In DNA, A is partnered with T and G is partnered with C, which remains true except that the T is being switched to U, uracil, in the RNA.
The RNA is copied one gene at a time. In the genes, there are combinations of nitrogen bases that signal the end and beginning of the gene. The parts we want to copy are called transcription units. In the beginning, there is a TATA box, meaning it has the nucleobases in the pattern TATA, AAA. And at the end is a termination signal, and that has the pattern ATAT, TTT. This signals to the polymerase when to stop copying. The part of the polymerase that senses the end is called SNRPs. SNRPs are nuclear ribonucleoproteins. The SNRPs and other proteins create spliceosomes that edit the copies to take away unnecessary parts. The parts that are left after editing are called exons, and the ones that are taken away to be recycled are called introns.
When the whole gene is copied, a so-called 5' cap made of guanine is put in the 5' end of the gene. On the other end, 3', we put a poly-A tail made up of adenine that makes it easier for the mRNA to be translated and transported. The end sequence then signals the completion of the RNA transcript. Once transcribed and edited, the RNA can be used as messenger RNA, mRNA, and is sent out of the nucleus. The mRNA must then find ribosomes in the cytoplasm or the rough endoplasmic reticulum so that it can be translated.